Today we are going to begin with the structure of the lysosome. And as you can see, it is that simple. It's not that complicated. Okay? It is just, I can say, like a sac. Okay? It's like a sac, like how you can see. And the sac has the space where the digestive or hydrolytic enzymes, they will be stored. And this is the matrix. So it is basically as simple as this. I think this is the simplest structure of an organelle within a cell. So these lysosomes, they are irregular, okay, spherical, and sac-like structure, like how you can see this. Okay, and within that structure, you have there diuretic enzymes, the digestive enzymes, and the sac, it is enclosed by a unit membrane, by a single membrane. Now this is not double membrane, and if you are to check its diameter, its overall diameter, it is roughly 1 micrometer. It is that small. Okay, now a single lysosome contains over 50 different enzymes. One single like this, you have over different types, over 50 different types of enzymes, but we can all call them the hydrolase enzymes. Why acid? Because they work in acidic medium. They work best in acidic medium. Okay, and actually there are these enzymes which are good. They are good. Why good? Because imagine all processes that occur in a cell. Okay, you have mitochondria, you have chloroplasts. They are formed and they are worn out. So you must be able to remove them. In immunity, you have their macrophages, these phage eaters, where these macrophages do engulf the pathogen. After they have engulfed, then they release these digestive enzymes, these lysozymes, to break down the pathogen into simpler molecules that cannot harm your body. And so they are so important in your body. Actually, not just important, but importanta. So this membrane, it is highly glycosylated. What is the meaning of glycosylation? You have there a lot of short carbohydrate molecules. And why should they be there? You know, carbohydrate molecules, they are receptors. They form receptors. Okay? And why do you need the receptors? Binding off or binding with a Golgi apparatus or with an endoplasmic reticulum. It is a receptor mediated process. Okay? So these membranes must therefore have receptors that will attach on the receptor of the endoplasmic reticulum or the Golgi apparatus, so that the process continues. Okay, so that depending on the process, otherwise whatever would come outside the cell would just bind, okay, with the Golgi apparatus, or would just bind with the endoplasmic reticulum, and it is transported within. But that's not how it is. You must have specific receptors. So it is highly glycosylated, forming a continuous glycoprotein layer, like how you can see this one. Okay, and the structure is like that of a mucin-like domain. Now we shall study this in chemicals of life. Okay, where we shall talk about these other polysaccharides, and we shall talk about mucin. Okay, that so this mucin is the one which resists the breakdown of the enzymes within the lysosome. Because imagine you have enzymes inside here, okay, and these enzymes, they are diuretic enzymes, too annoyed but you've packaged them inside this vesicle, inside this lysosome. How are they maintained inside without damaging the lysosome itself? It is simple. This membrane having that mucin, like, mucus, okay? Yes, there, how soft it is. Yes. So the mucus, it covers the interior, it lines the interior of this lysosome, okay? It's like the way you see the mucus that surrounds the the gut, the stomach, to prevent the action of pepsin. So it is the same, it's analogous to the same mucus which surrounds the sac of these lysosomes to prevent these enzymes from breaking the sac itself. Okay? Now if they, by mistake, they break this sac and they are released into the cytoplasm, just fwa. Okay? In other language you say just fwa. Then it becomes too much. However, because of that mucus layer that surrounds, okay, the membrane, then it protects them. It protects these enzymes from digesting the membrane so that they are maintained within the sac. Is that okay? Yes, I hope that's good. Functions, they do perform a range of functions, and we are going to see the functions. And one of the functions is autophagy. Autophagy. Auto means self, phagy means eating. So this is self-eating. Like, assuming you have a mitochondrion or any organelle, okay, that has worn out, okay, then you're going to remove that organelle by the action of these lysosomes. And the lysozymes in particular, whereby the primary lysosome, this one, okay, what happens first? The worn out organelle, okay, maybe the mitochondrion, maybe the chloroplast, maybe any organelle that is stored within the cytoplasm, in case it's worn out, it will be engulfed and packaged within a vesicle. And this vesicle is called a phago. This is an autophagosome, okay, not phagocytic vesicle. Phagocytic vesicle is the one which has been formed by phagocytosis. So this is an autophagic vesicle. So you can call it an autophagosome. So now you can see our organelles are fully surrounded within the vesicle. Now, after this process, after you formed an autophagic vesicle, then our primary lysosome is going to come and fuse with this vesicle to form something of this kind, form this stuff. As you can see, it's now a continuous membrane. Are you seeing? So when they fuse, then they will release these digestive enzymes, these lytic enzymes, these lysozymes into this vesicle, where they will mix up with these worn out organelles. And what will be formed is a new lysosome, which is a secondary lysosome. Out of this, and you can see how the mitochondria have been broken down. You can see, okay? Yes. So now this process is what we refer to as the autophagy. Okay? Autophagy. So that's simple. So digestion occurs by the lysosomal enzymes into the end products, and the soluble ones are absorbed back into the cell, and the non-soluble ones they'll be transported out, okay, by exocytosis. I hope that one is okay. Then another function is heterophagy. Hetero means different. With auto, it was self-eating. You were eating your own cells. Okay? But now with heterophagy, now you are eating outside components that have been formed by phagocytosis. I hope you can see this. Okay? So primary lysosome, they will fuse with a food virtue, okay, that is formed by phagocytosis. For example, when food is here, the membrane is going to invaginate, okay, and then the food or the microbes, they are surrounded within this depression, okay, and then after it will fuse at this point and form inside, and this is now a phagocytic vesicle. It has been formed by phagocytosis, endocytosis. Okay? Now after you formed this phagocytic vesicle, then you have your lysosome, okay, that has those lytic enzymes, the primary lysosome. It will come and fuse with our phagocytic vesicle to release those enzymes. And if the enzymes are released, the enzymes will break down this food, okay, so you form a secondary lysosome, and enzymes will break down the food into small soluble products that can now be absorbed into the cytoplasm. And those ones, the undigested ones, those large molecules that cannot be broken down, they will be excreted out of the cell's body by exocytosis, where it will fuse with a membrane, then the membrane evaginates, okay, and then closes off to release this vesicle outside. And basically, it is as simple as that. So what is responsible for that? They are these primary lysosomes. They are these tough, they are annoyed. Tough. So endocytosis forms a digestible virtue, which is a heterophagic virtue. We can call it a phagocytic. Hetero means different. Autophagic, it is auto because it had self organelles, the mitochondria, chloroplast, etc. But with heterophagic, it's because you have different organelles. Actually, not organelles. You have food materials from outside, okay, that have been formed by endocytosis. Then digestion occurs by the lysosomal enzymes, those lytic enzymes, and the products live by diffusion and they are absorbed into the cytoplasm with the aid of specialized transporters within this membrane. Okay, we have studied cell biology, the proteins within this membrane, and then those ones which are not soluble and digested, they are released outside the body by exocytosis. Then that was autophagy, but we also have autolysis. Now here, this is programmed cell death. Maybe a cell has turned to be cancerous. Maybe the cell has aged. Okay? The cells might end up divide, and somehow a cell misbehaves. Okay? The neighboring cells will release signaling molecules, okay, that will instruct that cell to undergo what we call programmed cell death. Okay? Apoptosis. Now, that cell is going to produce too much lytic enzymes packaged within the vesicle, then finally this lysozyme is going to rupture and release these lytic enzymes freely into the cytoplasm. Now these lytic enzymes, they will break down, they will digest all cellular components, everything in the cytoplasm. Okay? And the cell dies. So you have your vesicle that has proteases. Those are protein digestive enzymes. They will be packaged within a vesicle, and this vesicle is a lysosome. Okay? Now, since you have this cell is misbehaving and you want that cell to die, then you instruct the release of these enzymes, these proteases. And these proteases will accumulate in the cytoplasm and digest everything, break down everything that is made up of protein, okay, into amino acids. And now if you break down everything, now really will this cell really survive? So as a result, the whole cell will die. Okay? That's another function of these lysozymes. Then they also play an important part in developing process. For example, you know young toads, they have tails. The tadpoles, the newts, they all have tails. But mature toads, they do not have a tails. I think you are aware of that by now. So now where does the tail go? Where? And why does it go, by the way? Now the explanation for that, it can be like, it's simple. The mature toad does not need a tail. And actually, the tail might even disturb, might even make this mature toad to lose balance. Imagine a mature toad with a tail. But when it's young, remember it is jumping, swimming highly, okay? So it needs the tail to maintain that balance. But when it grows, the tail loses value. So it becomes a vestigial structure, okay? So it is going to be lost in the process of development. And there are these lysosomes that play an important part in the process of losing a tail. Okay? That when the right stage happens, then the cells are going to produce too much lysosomes that will digest the cells that attach this tail onto the animal. Okay? And when they digest these cells, then the tail will automatically clip off. Okay? And the mature one will remain without a tail. Okay? Yes. So it is as simple as that. Also in bone development, these lysosomes also play an important part in modeling the bones. So you study ossification in histology, and you good enough if you find there, you beat our. Also in fertilization, these vesicles play an important part. Actually, so much important part, or too much important part. Because in O level, when you are drawing a sperm cell, you draw something of this kind, something of this caliber, where this is the neck, okay, and then something of this kind, okay. This is, I think this is a centriole, this is the neck, and then you draw a tail. Okay? Now somewhere here you labeled an acrosome. And you said that that acrosome contains digestive enzymes. You remember that in O level? Yes. Now that acrosome is actually a lysosome. And those digestive enzymes are actually the lytic enzymes in the lysosome. Now, without this acrosome, trust me, the sperm cannot penetrate this zona pellucida. Now, I hope you've ever heard of that term. Without the acrosome, then the sperm cannot penetrate that zona pellucida. This layer here, this one, it is a layer of glycoprotein. Very many carbohydrate molecules within this layer that you cannot easily penetrate. It's not a membrane. Okay? You have this membrane, you have this zona pellucida, and then you have a plasma membrane, and then you have this layer here. Okay? This you first have these cells that they are called is it corona radiata cells? Yeah, these cells. You have these follicle cells, okay? And then you have the zona pellucida right from here, and then you have the cell membrane. So the head easily penetrates through the corona radiata. Actually, all sperm cells, all of them, they can easily penetrate through this corona radiata, that layer. But this zona pellucida, this layer, sperms cannot penetrate. So what happens when you reach? Now, should I talk about that right now? Okay, let me talk the science. These sperm cells, they do have receptors. Okay, they do have receptors within their head, and within this zona pellucida, this layer, you have their receptors. Okay, they are called, are they the Z receptors? I think they are called the Z receptors, something of that kind. Never, nobody is going to ask you. So when this sperm cell reaches the receptors on the sperm cell, they bind to the receptors, okay, of the of the of this ovum, and the binding of these receptors, they do depolarize the membrane. And when you depolarize the membrane, then you release calcium ions into this sperm cell, okay? And the release of the sperms of calcium ions into the sperm cell, it will induce the acrosomal reaction, which will release these lytic enzymes into this zona pellucida, where they will digest this zona pellucida to create that space, okay, for the head to be released into the cytoplasm. Now, once one sperm binds to the receptors, okay, of this ovum, it induces these other protein receptors throughout the ovum to undergo configurational changes, which will stop other sperm cells from binding to the receptor organ, ensuring that one sperm can fertilize with an ovum, hence preventing a condition known as polyspermy. So are you seeing the role of this acrosome? This acrosome, without this acrosome, then the sperm cannot penetrate. It cannot, I repeat, it cannot penetrate through this zona pellucida. It is hard. Okay? It's hard. So you bind, then you release, you cause some, you depolarize the membrane, you release calcium ions. Calcium ions induce the acrosomal reactions within the sperm, then you release the enzymes. Those lytic enzymes, they break down this layer, then the sperm cell gets way in. Okay? And then you fertilize. So these lysosomes, they do play an important part as far as fertilization is concerned. So acrosomes in the sperm releases enzymes that digest the lining, I think this is lining, no, the limiting membrane of the ovum to allow, Anita is going to comment on enable, sperm entry, and then initiate the process of fertilization. The lysosome in the cytoplasm of the ovum, they do digest the stored food, okay, to provide energy that is going to drive the process of reproduction, embryogenesis. Then we have the Golgi endoplasmic reticulum system. Okay, the Golgi endoplasmic reticulum, the lysosome system. They are these ones which work, okay, within this system. The Golgi body, those sacs that are formed, that internal transport, it is all about these sacs. So regulates the secretory activity of the Golgi apparatus and the endoplasmic reticulum, as well as the modification of the secretory products. So the whole of this system, okay, for you to pinch, if this is the nucleus, for you to pinch to move from here, okay, to the Golgi apparatus, you move through the vesicles. And if the vesicle contains lytic enzymes, and remember enzymes are formed from where? By protein synthesis. From where? From the endoplasmic reticulum. So if you move and you contain the lytic enzymes, then you are a lysosome. So you pinch off from the ER into the Golgi body, and then you pinch off. So the whole of this system, it works within that principle. Is that okay? In immunity, those macrophages, when a macrophage binds or engulfs a pathogen, you produce, I think I've already talked about this. You produce these lysosomes which will break down the pathogen into small substances, and they render it harmless to your body. So basically, these lysosomes, they play an important part within the bodies. Okay? Yes. Any question as far as lysosomes are concerned? Maybe let me check the chat box. So, like I said, we have these tiny sacs that also do perform specific functions within a cell. Okay? And these sacs, these particles, they are called microbodies. For example, you have their peroxisomes. Okay? And these ones, they do contain also enzymes. Okay? But now the enzymes, in this case, it is catalase enzymes. Okay? And the catalase enzyme, it is responsible for removal of hydrogen peroxide. So you can't just leave it within a cell, you also package it within a specific vesicle. Okay? And in this case, it is a peroxisome. So they allow a cell to get rid of toxic substances that are produced within a cell. And in this case, we have an example, hydrogen peroxide. Now, if this hydrogen peroxide, it is hydrogen, okay, with 2 oxygens. Now, this one is highly unstable, okay, highly unstable. That if it is not removed within the cell, okay, then it can end up oxidizing, okay, your DNA and other stuff, which will lead to altering the DNA. Okay, you alter the DNA, and if you alter the DNA, then that's what we refer to as mutation. Now, to avoid that, then you produce these peroxisomes that will always break down that will have enzymes that will break down. Okay, you produce peroxisomes. A moment. It's okay. So you produce these peroxisomes, okay, that will have the enzymes, and which enzymes? The catalase enzymes, okay, which will always break down. Yeah, it's okay. What I wanted is here. Yeah, it's okay. I've gotten it. Sorry, guys, you forgive me for a moment, but it's okay. So you have peroxisomes. Now, to get rid of hydrogen peroxide, you produce these peroxisomes, okay, that contain this catalase enzyme that will always break down the hydrogen peroxide the moment it is produced, so that you convert it to water and oxygen, which are neutral molecules. And in physiology, we usually carry out more of this enzyme. Okay, and I think you also did it in others, where you used any tissue. It could be liver, it could be tomato, and you put it in hydrogen peroxide. Maybe you are testing for temperature, you are testing for concentration, you are testing for surface area. You put it in hydrogen peroxide, and you are seeing effervescence, okay, of the colorless gas. So that was the catalase enzyme that you were dealing with. Okay? Yes, that's pretty good. And they are stored within these microbodies. Okay? Yes. So liver microbodies detoxify alcohol and fat soluble drugs. Yes, in the liver. Like I told you, I think you are the ones I told. Ethanol does not, and I repeat, doesn't have any nutritional value to your body. So when you take it, it must be removed from the body. So you have their microbodies within the liver that are responsible for removing the ethanol. Okay? Converting ethanol into non-toxic compounds. The drugs that you take, now here that's where you are concerned, I'm concerned with. During coronavirus, people used to steam. People used to take each and whatever kind of drug. So those drugs you took, they were, as a must, they were removed from the body. And what's that? It is the liver that is responsible for doing that. So in the body, in the liver, you have very many of these microbodies that will contain the digestive enzymes that will break down the fat into compounds, into metabolites, and that will break down the drugs into metabolites, which can be removed by the kidney, and you pass them out as urine. That's why when you want to confirm that, the moment you finish taking a drug, maybe you take excess, you pass out urine which is so concentrated. Sometimes too yellow, sometimes not understood. Okay? It is because whatever you take must be removed outside your body. So it is as simple as that. Then you have peroxisomes and lysosomes, are similar in appearance but differ in origin. Whereas lysosomes pinch off from the Golgi complex or Golgi body, peroxisomes, they do self-replicate, okay, that use proteins that are within a cytosol. Can you imagine? You form proteins within the for lysosomes. You form proteins and then you package these proteins, okay, and then you release them. It pinches off as a vesicle, containing lytic enzymes, and this one is going to be a lysosome. But for these microbodies, for them, they don't do that. They don't pitch off the Golgi apparatus, but they replicate themselves, okay, using the protein complex machinery that is found within the cytosol. Okay? Yes. And examples, we have the glycosomes, okay? And these ones, they do contain the enzymes that are responsible in breakdown of lipids or fats during seed germination. And with germination, you know there is a lot of physiology. The process of germination is not just a joke.
Teacher,
Yes,
teacher, so the glycosomes are not found in the human cells?
most probably they are not found there. They are all the same, but just the nomenclature is the one that varies. Okay? And this one, I'm sure it comes from. Okay. Okay, we can say they are not found in human cells. However, they are the same. Identical, everything is the same, just the difference is nomenclature. Okay? Good. So any question as far as those ones are concerned, the microbodies are concerned? So let's talk about the cilia and the flagella. Now, like you're seeing, one is flagellum, many flagella. One is cilium, many cilia. And these are structures, I can say the extensions of the plasma membrane that are responsible for locomotion, performing different functions as we are going to see. Okay, they do perform a diverse range of functions. So cilia and flagella are structurally identical, okay, but they have similar structures as we are going to see, but usually cilia they are numerous, very many, like how you can see, okay, and they are short, small, slender, but flagella they are few but elongated, but they perform relatively similar functions. However, they also do differ, as you can see this. So if you have to compare, like I've told you, cilia, they are usually numerous. Flagella, they are less in numbers. They are short, hair-like organelles of about 10 micrometers. But these ones, they are long, whipe-like. You can see. You know a cane? You know a wipe? If I'm to wipe, if I'm to wallop, if I'm to argue, especially using that wire. You know how it feels? So this is occurs throughout the cell surface. It is present at one end, like how you see. This one is throughout the entire body surface. But this one is just at one end. They do beat in a coordinated manner. These many cilia, they beat in a coordinated manner to bring about movement. Like if they start to beat from here and they are all beating from this side, then they will all beat, okay, in a coordinated manner. But for this one, just beats, just whips, okay, it just whips, and the whipping creates that thrust force, which is going to push the organism forward. Show sweeping movement or pendular stroke. Sweeping movement, it's like if they all bend, then they'll all bend, okay, they all bend in that coordinated manner to move the material across. But for this one, it just beats, it is undulatory movement, like how a snake is moving. It moves So that's that beating. It's the one that we refer to as ambulatory. Okay? Yes. However, regardless of those differences, they do have similar structure. Okay? And if you are to describe the structure, that is a structure. So they do arise from granular structure called a basal body. So they do all have a basal body. This one, it is a basal body. Okay? It is a basal apparatus. And the reason why I say it is an extension of the membrane, you have there macrotubules or microtubules and microfibrils, but all those microtubules, they are enclosed within a membrane, within a cell membrane, like how you see here. If this is your cell membrane and this is your cell membrane, it encloses these microtubules, okay, to form what we call a flagellum, to form what we call a cilium. Okay, so covered by a unit membrane, yeah, the same, I've talked about that. It is an extension of the membrane. Central filament is called an axon. This one, okay, it is an axon. And when you try to study the structure of the axon, that's what you see. Where this is the membrane of ours, okay, and then these are the microfilaments, okay, there are 9 of them, and these are the macrotubules. They are 2 of them. So in some books, not some books, even here you find 9 plus 2 arrangement, and I'll be able to show you what that 9 plus 2 arrangement. So they are formed from 11 macrotubules, okay, and the 11 macrotubules, they are in this pattern. They are in 9 plus 2 arrangements. Those are the 2 in the middle, these doublets, these doubles in the middle, and then the 9 doublets that are peripheral. You can see this is 1, 2, and then this is 1, 2, 3, 4, 5, 6, 7, 8, 9. Okay, so that's what we refer to as 9 plus 2 arrangement of microtubules, and it is the same in both cilia and flagella. Okay, and there are 2 central singlets. Yes, these ones, they are single, they occur in singles, and then the 9 peripheral doublets. These 9 ones, they occur in double, and there are 9 doubles, okay, of macrotubules that span. Now this is a cross-sectional area. When you cut from here, this is what you see. When you cut from here, this is what you see. But when you cut from here, okay, at that stalk, at that point, then this is what you see. Now here you won't have those 2 singlets, and it won't be covered by a unit membrane. Note, each centriole is made up of 9 triplets of microtubules arranged in a 9 plus 0 arrangement. We are going to see the structure of a centriole. So are these cilia important? Okay, you can write some. Write some.
Teacher, the network is bleaching. We can't see some notes very well.
Oh God, sorry. What is wrong today? Yeah, your functions are good. Your functions are good, but they are more than that. Okay? You could write more. Dry food into its gullet, its oral groove, its mouth. The mouth is called not oral groove. I think. Okay. Okay. Into its gullet, its mouth. Okay. And the cilia which beat rhythmically. You saw the whole body is covered by cilia. So they do beat rhythmically to bring the food directly to the oral groove, to the mouth. Okay? Then they facilitate gas exchange by passing water currents to gills in certain water cells. Then in echinoderms, for example, starfish, the ciliary movements do allow locomotion by driving water through its vascular canal system. Now, these molluscs, these echinoderms, they have a vascular canal system. Okay? And for them to locomote, this vascular canal system performs both locomotion and nutrition. The vascular canal system performs an important part, where the cilia direct a flow of water current to this canal system, where they will absorb the food. If food is, they will absorb the food, digest and get food, but also this flow of water current to contribute to the locomotion of these organisms in water. Then respiratory tract, your trachea is fully covered. It has those tuboidal epithelial cells that are ciliated even. So now the cilia, it's the one which beats rhythmically to remove the trapped particle from where it has been trapped, and it brings it back to the surface, where you're going to either cough it out or you cough it and swallow to be digested. So it is the cilia that performs that. It is the cilia that performs that. Then the oviduct, the fallopian tube. Yes, the fallopian tubes, they move the ovum from the ovary to the oviduct, where fertilization is going to take place. Even after fertilization, the cilia will keep moving that oviduct, and the ovum, the fertilized ovum, which is the zygote, to the oviduct, where they will implant, and the process continues. Then cilia in the nephridae of annelids, they play an important role as far as excretion is concerned. Then sperms, yes, sperm cells, they do have the flagella, okay? And there are these flagella that beat rhythmically to move the sperm cell to provide that propulsive force, which is going to move the sperm cell towards the ovum. Then, locomotion in certain protozoans, such as Euglena, Chlamydomonas, Trypanosome. Those ones, they do have flagella that beat rhythmically to bring about that propulsive force, which is going to move the organism forwards. And so on, so on, so on. Then you have the cytoskeleton. This one is a system of macrotubules, or it's a network of fibers.
My brain is on fire.
Why? Okay. Yes, Santa.
No, let's continue.
Skeleton. And here I'm going to go at a slow pace. Okay? I'm going to go at slow pace. Now, like I told you, I still take you back to Form 1, where you drew such a cell, and you say this was a nucleus, and then this is a cell membrane. Now, you thought this was just space, really? You see that you were disappointed for no reason, or they lied to you, and then they say this is nucleus, this is cell membrane, and then this is cytoplasm. So you thought cytoplasm was just an empty space? No. the cytoplasm actually is not just an empty space. And if I still take you back to the other first peak of ours, you remember the very first lesson? Actually, let me even take you there. So this is the peak that I'm talking about. This was our first, okay? And you can see. So if this is the nucleus, it is this tight, okay? It is this tight. This is just the cytoskeleton, okay? It's a network of fibers, and there are those fibers that do contribute to the overall structure of a cell. Even when you zoom in, this nucleus, it also has very many fibers. Very many fibers where the chromosomes will attach, in fact. Okay? Somebody has just joined. I don't know who is she or who is he. So you have very many fibers that contribute to the cytoskeleton. Okay? And they are actually these fibers that attach on the protein molecules within the plasma membrane. Okay? The membrane is not shown. They attach on the proteins within the plasma membrane for anchorage, so that they support the cell to remain an intact structure. Okay? So that is the cytoskeleton for you. Now, so that's our cell. Okay? That's our cytoskeleton. So it is a complex network of fibers that extend throughout the cytoplasm. Some attach on the proteins within the plasma membrane, but they all originate from the centriole. Okay? And that's where they attach. In animal cells, plant cells don't have a centriole. And it is that complex network which maintains the overall structure of a cell. And it organizes the cell structures and activities, anchoring many organelles, okay, in the cytoplasm. So they are not just suspended in space. They have points of attachment. They have where to attach from. Okay? And so these many fibers, okay, within the cytoplasm, they are the ones that do contribute to the cytoskeleton. And they occur in three types. Okay? One, we have the microtubules. So this one came first, which is okay. So we have the intermediate filaments, and I'm going to say why they are intermediate. Then we have the microtubules. Okay? Now these ones, they are thick, or they are the thickest of all the three. And their diameter is it ranges, is it from 50? Not 50, the 50 is too much. From 25 micrometers. Then these microfilaments, for them they are tiny. We are going to study each, and you see the structure. These are tiny filaments, tiny fibers within the cytoskeleton, within the cytoplasm. And they also do contribute to the cytoskeleton. Then you have intermediate filaments or intermediate fibers. Now for these ones, they are in between these two, basing on their diameters. Okay? The diameter rise. They are not microfilaments, neither are they the microtubules. Okay? But they are intermediate. So the name, intermediate filaments. Now what are microtubules? These ones, they are hollow, cylindrical rods of about 25, okay, in diameter, and about,
Teacher,
yes,
a question about the previous slide.
yes, a question, good, ask.
So, are you trying to say that the cytoskeleton, it is also divided into these microtubules, the microfilaments, and intermediate filaments and stuff?
Yeah, that's it.
Or they
So
are just types?
okay, let me answer that. So somebody is asking that the cytoskeleton is divided into microtubules, macrofilaments, and intermediate filaments. Now they are types of fibers that constitute a cytoskeleton. Types of fibers that constitute the cytoskeleton. And those ones which are cylindrical, large in size, they are microtubules. They are tube. You know a tube? A tube, it is hollow. Okay? So they are hollow even. These ones, they are also hollow. And they have a large diameter compared to the three. Then the micro, these ones, they are tiny, actually made up of just two polypeptide chains twisted together into an alpha helix. Okay? We are going to see. Then you have the intermediate filaments. These ones now, they are not tiny, they are not small, they are not big. They are just in between the microtubules and the microfilaments. So all of these three, they constitute a cytoskeleton. And depending on which cell, depending on the location of a cell, it will determine which one will be prominent. For example, the intermediate filaments. You have, is it desmin? We shall find it down. They are abundant in muscle cells, okay, where they contribute to the muscular contraction. You have the keratin. It is an intermediate filament. Depending on the type of keratin, you find it in hair and also in fingernails and in the underlying skin, because the skin is simple squamous keratinized epithelium. So that keratin, you find it there. So they do contribute to the cytoskeleton, okay? But they are three. They occur. These fibers occur who can be classified into three classes: the microtubules, the microfilaments, and the intermediate filaments. I hope that one is okay. So let's talk about the microtubes, the tubes. So these are hollow rods of about 25 nanometers in diameter. From here, okay, to here. The diameter, if this is a tube, if this is a hole, the diameter is this. Okay, and it goes like this. So that's the diameter. So for them, they have about 25 nanometers of diameter. And they are about 200 to 25 microns long, their length. Okay? So in other words, they are elongated within the cell. Okay? And you can see it is just a fibrous protein made up of two polypeptide chain, the beta tubulin and the alpha tubulin. These two. And these polypeptide chains, they are the ones that are wound together into an alpha helix, into a helix, to give rise to a tube, like how you see. Okay? So the function, they provide shape of the cell, the gliding movement of organelles to be able to slide. Okay? They slide over. In other words, they provide that point of anchorage of organelles. The cytoplasm is not just space. Okay? So they provide that surface over which organelles they will freely grind. Okay? Then separating chromosomes during cell division. You know, spindle fiber, they do form. And so what are the spindle fibers? What are they? They are the microtubules. So they are the ones that will separate the chromosomes so that each chromosome or each chromatid goes to its individual cell. Okay? In cell division. So basically, that's how it is. Then, the functions, I think I've repeated this slide, but it's there. Moving other organelles through the cytoplasm, major component of the cilia and flagella, which are important in cell motility, form spindle fibers during cell division, shape and mechanical support to a cell. And where do you expect to find it? Found throughout the cytoplasm of all eukaryotic cells, and they form part of the cytoskeleton that gives rise to structure and shape of the cells. Is that okay to everyone? Anita and Edwin? Is that okay? Yeah.
Teacher, before we come to the microtubules,
Yes. So let's talk about microfilaments. We shall talk about the centrioles later. So now these microfilaments, they are also part, they are constituents of the cytoskeleton. However, for them they are small in diameter as compared to the microtubules. So these ones, they are solid rods of about 7 nanometers in diameter. Okay? And they are built as twisted double chains of actin subunits. So all these are fibrous proteins. Okay? They are all proteins and they are all fibrous proteins. However, the difference is these ones, they are tiny. They are small. Okay? And they are also highly twisted actin subunits. Okay? To form what we call the microfibrils or the microfilaments. Okay? So you get the difference between the tubules and the microfilaments. A filament, it's like a fiber, a thread, tiny. But a tubule, it's like a drinking straw. Okay? So the other ones, they are even hollow. Okay? With two alpha chains, alpha helices, actually with two helices. The beta tubulin and the alpha tubulin. But now with this one, it's not hollow, they are small it's like thread-like fibers, very small. Okay? With also two chains, two polypeptide chains that are twisted together. Okay? That are highly twisted together, like how you can see here. Okay? Like how you can see here. This is, I think, the space feeding mode of the actin filaments. So the structure, the structural role of microfilaments is to bear tension and resist pulling force within a cell. So in the case of any tension that is created within a cell, okay, then the cell does not overstretch. Okay, so these ones, they are responsible for making cells maintain their own shape and resist expansion. They form a 3D network called a cortex just inside a plasma membrane, which supports the cell shape. The cell shape. Bundles of microfilaments make up a core of microvilli. in the intestinal cells. So since they are tiny, they are the ones that make these microvilli to remain standing, not to collapse. Okay? Remember, they are fibers.
Excuse me, sir.
Yes, Edwin.
What does it mean by a 3D network?
A 3D network. Okay, somebody is asking 3D network. Now, 3D network, I think, in simple terms here, we are to form that overall structure. Now, a 3D picture is the one which you can view the both sides. Or 3D, you can view both sides. Okay? Then 2D, you just view, you just have a plain view. So within the membrane, you form that highly coiled 3D network of actin filaments, okay, that maintain the cell structure or the cell shape, okay, so that the cell does not easily collapse, the cell does not easily burst, because we have seen that there are a lot of things within the cell, there are a lot of contents within the cell. So these actin filaments, they are the ones that maintain the overall framework of a cell, okay, and we can see they are tiny fibers, okay, they are tiny fibers. Then with intermediate filaments functions, dividing cell membrane pinch off into two cells. So they also play part in cell division, involving cell movements, e.g. amoeboid movements, phagocytosis and pinocytosis. So these microfilaments, they play an important part in amoeboid movement. You know amoeboid movement, it involves converting the cytoplasm from one form to another form. Now when you convert from one form, which is a jelly form, to another form, which is solid form, then the membrane, in order to resist overstretching, then you'll be having these actin filaments, okay, that will be able to allow the membrane to extend a bit, but not to over extend and rupture. So that's why they are involved in movement. We shall study this amoeboid movement in locomotion, not now. Then muscle contraction. Yes, these actin filaments and myosin filaments, they do perform a strong function. They have a big role as far as muscle contraction is concerned. We have the sliding field of muscle contraction, and can we talk about it now? Or it might be a long process. But the actin filaments, which we have seen, they are the ones which actually slide over each other, okay, to bring about the muscle to contract, to reduce in size, hence contracting.
Teacher, previous slide, please.
Somebody wants a previous slide. It's here.
Done.
Okay, the previous slide is done. Then support the cell membrane and maintain the cell shape. What do you expect it? They nucleate at the plasma membrane with the cell periphery, or at the edges, having the highest concentration in those periphery ends within the plasma membrane. So that's where they are abundant. And since they're involved in maintaining the cell shape, then you expect them to to become abundant within the cell membrane. Then with intermediate filaments, now these ones intermediate because of their diameters, it falls between the small actin filaments and the large microtubules. So that's why they are called intermediate filaments. So their diameters, they fall within this range, 8 nanometers to 20 nanometers. And we saw that with actin filaments, they have a diameter of 7 nanometers. And with the microtubules, they have a diameter of 25 nanometers. So now for these ones, they do fall within that range. And what do they do? For examples, we have keratins, which are found in animal epithelial cells. We have desmins. These are found in muscle cells, okay? And they are responsible for muscular contraction. They do integrate the sarcolemma, the Z discs, and the nuclear membrane in the sarcomeres of muscle cells. Now I cannot talk about each here, okay? I cannot talk about each here, but we shall meet this in the promotion. Then we have peripherins. Now these ones, they are found in neurons, okay? Peripherins are neurofilaments. They are found in neurons. They are the ones which maintain their shape, the fibers not collapse. Then we have the laminins. The lamins, they are found inside the nucleus. Now these ones, they do provide the points of anchorage of the chromosomes within. They are the ones which actually responsible for the nuclear skeleton, to provide the overall skeleton of the nucleus inside, and the anchorage points within the nucleus. So they attach chromosomes to the nuclear membrane and provide anchorage points for the nuclear pores. Functions: tension bearing and maintaining shell shape and rigidity, anchor in place several organelles, including the nucleus and then the desmosomes. Now these ones, they are junctions that are formed between the two epithelial cells. Two cells, they must be anchored together. So they are anchored by at least. And the desmosomes, you have their surfaces for these intermediate proteins. So they attach their cells from different, the filaments from different cells, they attach at those junctions to maintain the two cells closely attached together. We shall study this in histology and see. Then, formation of the nuclear lamina, that is the nuclear skeleton, a net-like or a meshwork array that lines the inner nuclear membrane and governs the shape of the nucleus. And then finally, you have a loop of this nuclear lamina. So, if this is the nucleus, this is the nuclear pore, okay? You have the complex protein that crosses the pore. Then these fibers, these strands, okay? They are our intermediate filaments that are found within the nucleus, where our chromosomes will attach, okay? Yes. Otherwise, if you don't have them, if it is just a space, then I don't know if you can. Then finally, we have the centrioles or the centrosome, okay? The overall complex, if it is one like this, if it is one, it is called a centriole, okay? But if there are many, the whole complex is called a centrosome. And now this is like the center where all these microfibrils and microtubules and the intermediate filaments, they will all assemble from. they will all organize themselves from, they will all originate from, for the case of animal cells. But for plant cells, for them, they don't have centriole. So in many cells, the microtubules grow out from the centrosome near the nucleus. So it is the microtubule organizing center within a cell. So I think you can now see how important these structures are. And in animal cells, for instance, you can see you're going to originate the microtubules from these centrioles, okay? And the microtubules will attach chromosomes, and you separate the chromosomes. So the centriole has a pair, the centrosome has a pair of centrioles, and each with 9 triplets of microtubules arranged in a ring. They are the ones which will elongate, okay, and attach at the opposite poles, okay, to bring about separation of the chromosomes during cell division. So like I said, this will be the end of our topic, cell biology. And we shall begin with a new topic, which is chemicals of life and sea.